11. We'll do it like first thing. You kind of know what to expect, so all that stuff. So here's the stuff from today's lab. I'll go through, I have a pretty good model of the lymphatic system and immunity, so I'll go through that model. Lymph node I do not have a model for, so I'll have to use a diagram similar to what's in your PowerPoint, but I'll go through a picture of that.
No, I have my labs on today.
Spleen, I have a good model of the spleen, so I'm okay there. Lymphatic nodules, I have the sagittal face model that shows the tonsils. That's pretty good. I don't have any model that shows the Peyer's patches, but I'll show you a picture of that. Antibody, I have a picture but no model, so I'll go through that as well. Then I'll go through those microscope slides. So let me go through then this big model that's here that shows like the whole lymphatic system, or most of it.
How do I do that?
Okay, so all right, so this thing that's down in the abdomen, anyone know what that's called? That's the cisterna-chyli, all right? That's kind of the collecting area for all the lymphatic fluid that's in the abdomen, that's in the lower extremities. It all has to stop off at that cisterna-chyli, so that's that big kind of nodule there. And then directly connected to this cisterna-chyli is the thoracic duct, which is this guy that's going up the side here. You can call it the thoracic duct, or it's also known as the left lymphatic duct, so it goes by either name. But anyway, if you follow that up, it's going to empty into the left subclavian vein up there. So that's the thoracic duct. On the right side, there's a much shorter right lymphatic duct that empties into the right subclavian. So let me go up and give you like a picture of this stuff. So there's the thoracic duct going up and over into the left subclavian. This little guy that's really short, that's the right lymphatic duct. Now, whether you're looking at the right lymphatic duct or the thoracic duct, there are three trunks that empty into it. This trunk that goes out that way, that's called the subclavian trunk. This trunk that goes up towards your neck, that would be the jugular trunk. And then this trunk, which if you follow it, it actually goes down and then into this area, that's the bronchomediastinal trunk, okay? So that's on the right side. On the left side, in some ways a little harder to see, there's the subclavian trunk is coming in right there. The jugular trunk is going up there. And then if you want to see the bronchomediastinal trunk, you actually have to kind of tilt this model to the side because it's like hiding behind here. And it looks like this. You see it coming in there. It's sort of sneaking its way in and then attaching up there. So that's the bronchomediastinal trunk that's coming in there. Other stuff you can see on this model. Back this up. You have a number of lymph node groups. So these are the cervical lymph nodes, obviously in the neck. Underneath the chin would be the submandibular lymph node group. The armpit, of course, is the axillary lymph node group. The group that's in, this area would be the bronchomediastinal lymph node group. And then as you go down here, that's going to be the inguinal lymph node group. Okay. That's most of the stuff that you can see on that big model. Some other stuff that's mentioned there in that little section, lymphatic capillaries. So lymphatic capillaries, they're obviously too small to see on this model, but there's a decent picture from your lab guide, or sorry, not from your lab guide, from your textbook. This is a picture of some lymphatic capillaries, right? So the only thing to tell you about that is, remember, they're small. The key feature is they're closed at one end, right? A blood capillary is not going to be closed at one end. Another key feature are the anchoring filaments that act like little ropes that kind of pull on the walls to create bigger openings so the lymphatic fluid can then move into them. The next thing that's listed here is the thymus. The thymus, we literally only have that one model that has the thymus on it. And this is the model I'm speaking of. So, right? So this is the thymus here, sitting on top of the heart. Actually comes off in this model. So it sits sort of superior and anterior to the heart. This is probably from like a child, right? Because as you get older, the thymus gets a lot smaller, right? So it's pretty hard to see, but that's the thymus there. Next thing in the lab guide is the lymph node, and there is no good model for the lymph node, so once again I have to go back to a picture. Find that. So here's a picture of a lymph node, and anyway, so surrounding the entire lymph node on the outside, that would be the capsule. Connected to the capsule and helping to sort of organize the lymph node are these little branches called trabeculae. They're just there as little dividers to kind of divide it. And you're gonna, in all, like the thymus and in the spleen, you're also gonna see like a capsule and trabeculae when you look at the thymus or when you look at the spleen or you look at lymph nodes. It's a way of just sort of organizing them. Other stuff that you can see on here, you got these different vessels, right? These vessels that bring the fluid in, those are the afferent lymphatic vessels. And there's usually going to be several of those. And then the ones that bring it away, and there's usually only one or two, those are going to be the efferent lymphatic vessels. When you have a solid organ and there's an area where a lot of vessels, blood vessels, lymphatic vessels, airways connect, when there's like one area where all that stuff connects, they'll usually call that area the hilum. So on a lymph node, this area here is called the hilum. Okay, it's just the spot where the efferent lymphatic vessels are attached. You're going to see the word hilum a lot in A&P2, like you'll see it again when we talk about the kidneys, where like the blood vessels in the urine are attached to the kidneys, that's the hilum. Or where like the bronchi and the major blood vessels connect to your lungs, they call that the hilum. Okay, it's a word you should get comfortable with. This whole area on the outside here is the cortex. And these sort of roundish structures you have out here, those are collections of lymphocytes. That's why they're colored purple. Remember, one lymphocyte has a really big purple nucleus. So when you put a bunch of lymphocytes together, it's going to create areas of purple. The name for these, they call them the cortical nodules. And inside of the cortical nodule, it usually stains a little bit more clearer, and they call that the germinal center of the cortical nodule. This area is going to be the medulla. When you have lymphatic tissue in the medulla, like here or there or like there, they call those medullary cords. Versus all the other spots that you see kind of filling in here, those are all open spaces where the fluid passes through, and they call that the medullary sinus. Okay? So all the fluid's coming in from the outside, it trickles through these different sinuses, kind of towards where the medulla is, it collects there, and then it moves out using the efferent lymphatic vessels. All right, I think that's most of the stuff to show you on the lymph node. Next we have a spleen, and we do have a decent model of the spleen. Lose weight. All right, try to gain weight. All right. So here's our model of the spleen, right? This is kind of the anterior view of the spleen. Couple things to notice. This guy in red is going to be the splenic artery that's coming in. If you flip on the back side, this guy in purple is the splenic vein that's carrying the blood away from the spleen. If you go back to the anterior view, this area here is kind of concave, and that's because the stomach would be sitting kind of on top of it and pressing into it. So they call that the, because of the stomach being there, they call that the gastric surface or the gastric impression. If you flip the spleen over on the back side, there's an indentation there as well. That's where the left kidney would be pressing. So they call that the renal surface or the renal impression. All right. Now, for lymphatic nodules, that's the next thing listed. We do have this sagittal face model that shows the tonsils. This is a pretty good model for the tonsils. We don't have a good model, though, for Peyer's patches, so I'll show you a diagram of that. Anyway, when you look at the sagittal face model, there are three types of tonsils that you have. This one, and for whatever reason on this model, they kind of use this sort of orangish color for some of them, but then they didn't do it for the other one. So this thing at the back of the tongue, that would be the lingual tonsil. This thing that's kind of on the side of the soft palate, that's called the palatine tonsil. And there's going to be a left and right palatine tonsil. And those are the things that, in people that have a throat infection that can get really big, like if you look at someone's throat and they've got enlarged tonsils, it's these palatine tonsils, and they can actually sort of touch across the midline when they get really enlarged. And then the last tonsil that you have is this guy up here. That goes by a couple of names. They call it the adenoid, or it's also called the pharyngeal tonsil. So the purpose of these tonsils, they're strategically located so that food or air that might be contaminated with bacteria or viruses, as it's coming in, it's going to press against these tonsils, and if there's anything there, the lymphocytes that live inside of these tonsils can start to mount an immune response against the bacteria or the virus that was inside of there. That's kind of, so they're there for a reason. They're there because they represent the entranceway into your digestive tract and into your respiratory tract, right? When people have a tonsillectomy, they usually take out this one, the palatine tonsils, and then they take out this one, the pharyngeal tonsil, or the adenoid, okay? They don't take out the lingual tonsil. Usually it doesn't cause any problems and it bleeds a lot, so they don't usually take that one out. All right, Peyer's patches then. And I mean, if you were to make a microscope slide of these tonsils, you'd see a lot of purple again. There'd be lots of lymphocytes kind of clustered together, you know, just like what you saw in the spleen or what you see like in your lymph nodes, same type of deal. Now for the Peyer's patches thing, Peyer's patches are collections of lymphatic nodules that are basically located in your small intestine, mostly in the last part of your small intestine, which is called the ileum. Okay, there's 3 parts to your small intestine. When you leave the stomach, you go into the first part, which is called the duodenum. Then after that, you go into the second part, which is called the jejunum, and then finally you go into the third part, which is called the ileum. If you did a cross section of the wall of the ileum like this, you'd see these kind of round purple structures, and those are the lymphatic nodules that are referred to as Peyer's patches. Okay. Again, I don't really have a good model to show you on, but it's there. Again, it's there for a purpose. You have nodules also in other parts of the small intestine. It's just that in the ileum, there's a lot of them, and there's, you know, they decided to give it a name, but they're all there for a strategic purpose, so that when you absorb food, if that food is contaminated with bacteria or viruses, hopefully it'll be put into contact with the lymphocytes that are in these nodules, and then that will mount an immune response. Okay. All right, so those are Peyer's patches. Next we have antibodies. And I don't have any good model of an antibody, so this is the picture from your PowerPoint. Just to refresh your memory, the antibodies are, this is a IgG type of antibody. It looks like the letter Y. It's made up of 4 polypeptide chains. This one in red and this one in red, those are called the light chains. And then the one in yellow and blue are the heavy chains. The chains are all held together with these things. These SS, that stands for those are the disulfide bonds. Okay, that's holding them all together. Remember I told you in lecture, this part and that part are going to be the parts that change, and they're going to be specific for a particular antigen. So those are called the antigen binding sites, or they're also known as the variable regions because they change. The rest of the antibody part doesn't change from one antibody. It stays the same, and they call that the constant region. Microscope slides. So this is the thymus, and you're going to have some terms that you already heard. You're going to have a capsule. So this is the capsule around the outside here. Connected to the capsule are the trabeculae. So those pink things that kind of are working their way in, that are dividing the thymus up, those are trabeculae. So you have, again, those terms when you're looking at lymph nodes, when you look at the thymus, when you look at the spleen, you have a capsule and you have trabeculae. Now, one thing that's weird about the thymus that's different from the other things that you're looking at is how they use the words cortex and medulla. For most organs that you've encountered, the entire outside area would be the cortex, and then like the middle area would be the medulla. But that's not how it works with the thymus. With the thymus, first of all, you have these structures that are called thymic lobules. So if you look at this diagram, each one of these little structures that you see, each one of these would be a thymic lobule. Okay, they're kind of formed by the trabeculae that divide it up. So each one of those things is a thymic lobule. When you look at one thymic lobule, that's when you see the cortex and the medulla. So for example, if you look at this thymic lobule here, the outer part that's a little bit darker would be the cortex, and then the inner part that's lighter would be the medulla, right? Or if you're looking at like this one here, right, the outer part would be the cortex, and then the inner part that's lighter would be the medulla. So it's a little bit different way that they name the cortex and the medulla. They base it on the lobules. Here's a closer up picture of a whole bunch of thymic lobules. You know, each one of these things would be a lobule. The darker part on the outside is the cortex, and then the lighter part that's on the inside would be the medulla. Now, when you look at the thymic lobules, again, to continue kind of that analogy with school, the fresh, new freshman T cells, they're basically coming into the cortex part of each lobule. And then as they get their schooling or their training, they're moving towards the medulla. And then the graduates are going to be found, you know, somewhere in the middle of the medulla. The losers that don't make it, they leave behind these tombstones that are called Hassel's corpuscles that kind of look like this. Or they're also, you can also call them thymic corpuscles. So you'll find these guys only in the medulla part of each lobule. That's where you look for the Hassel's corpuscles. Okay? They sort of kind of look like a cut onion. I mean, it can sometimes be hard to tell this apart from like a central artery in the spleen. All I will say is the central artery usually has a hollow lumen on the inside, whereas these, it doesn't really look hollow on the inside. Here's another one. Again, I don't know, maybe it looks like a cut onion a little bit. Or whatever. What did it look like?
Like a rose.
Looks like a flower. All right. We have some like George O'Keeffe fans here. Looks like a vulva. No, I'm just kidding. All right, sorry. Doesn't look like a vulva. Sorry. All right. Looking at the this is a lymph node. Okay. Now lymph node, once again, it's going to use that more conventional way that we talk about cortex and medulla. So this entire dark purple area on the outside, all around it, would be cortex. And then this lighter part in the middle is going to be the medulla. Okay. Once again, on the outside, kind of this tissue, some of it's pulling away here, but that would be part of the capsule. You can see a little bit of it up there. Some of the darker pink areas that are kind of working their way in from the capsule, those would be trabeculae. All of these round structures that are out in the cortex, those are cortical nodules, and the lighter staining part inside would be the germinal centers. Okay? Again, these are big collections of lymphocytes. That's basically what are causing all of these purple areas. When you have lots of lymphocytes together, they're going to look purple. Okay? Now, on this particular one, the hilum is probably down here, and you probably have the efferent lymphatic vessels coming off, although it's kind of cut off here, you really can't see them. The afferent lymphatic vessels are going to be around the outside of the cortex. They're usually really hard to identify, but like maybe this is a little afferent lymphatic vessel coming in down there, or I don't know, maybe like another one, little one over here. They're a lot smaller usually than the efferent. Here's another one. Again, this one I think shows some of the trabeculae a little bit better. So like you can see a trabeculae kind of working its way in up there and working its way in there and over there a little bit. You can see the capsule going around the outside. All of this is cortex. The round things are cortical nodules. The lighter staining part on the inside is the germinal center. This is all medulla here. And then down in this area, that's where the hilum would be. This is actually part of an efferent lymphatic vessel. I know it doesn't look like much. As for the afferent lymphatic vessels, it's kind of hard to see anything that's really jumping out at me. I mean, that might be a little one over there. They're tricky to find. It's nice when they label it, obviously. So this is the cortex. It's almost overstained. It's kind of hard to really even see the nodules. It just sort of looks like this sort of darkish purple on this one. There's the hilum region. Now they're using the word hilus because that's the plural form. I don't know why they didn't use the word hilum here, but when you have two or more hilums, it's hilus, okay? Those are the efferent vessels there. And then this guy up here would be like an afferent vessel that's kind of coming in. That's actually a fairly large one. I'm surprised they found it, but anyway. Then this is a lymphatic vessel, and you can kind of make out sort of this light colored structure that looks sort of like an upside down V. That's the valve that's inside the lymphatic vessel. This is a much better picture showing the valve. You can see it on the inside there. This then is the spleen, and a couple of things to notice, like on this at the top up here you can see a little bit of the capsule, and then kind of the solid like purplish, light purple that kind of coming in, like you can see it here, or you can see it going across there. That would be trabeculae. That's some trabeculae coming across there, like a little bit of trabeculae down there as well, just helping to divide it up. Everywhere you see purple blotches or blue blotches, whatever you want to call it, those are going to be areas of white pulp. Okay? So the purplish circles, that's all white pulp. Everything else that's filling in the spaces and the gaps, that's going to be red pulp. Okay? Red pulp is where the red blood cells are. It's where platelets can be stored. White pulp is where you have collections of lymphocytes. This one's maybe a little bit more subtle, but if you squint a little bit, you can make out areas that look a little bit darker blue or darker purple. Those will be pockets of white pulp, and then the intervening areas are going to be the red pulp. This is a good one showing the capsule and some trabeculae. So there's the capsule up at the top. This is a trabeculae that's kind of cutting its way through. There's a trabeculae coming in from there, trabeculae coming in from there. It's a little harder to see like the white pulp and the red pulp, but that, that's probably a little bit of white pulp there, like up there. Most of the rest of that down there. Most of the rest of it looks like red pulp. Now this is a super duper close-up showing you, here is white pulp. Most of the rest of this is red pulp, except like this is a little bit of trabeculae, it looks like coming across there. When you're looking at the white pulp, someplace inside of the white pulp there should be a little blood vessel, and that's called the central artery. It may or may not actually be in the middle. It just depends on if you get lucky in how they cut it. Like, don't expect this to necessarily be right in the middle, but it's usually located someplace within the white pulp. Okay? Every bit of white pulp should have a central artery located someplace. This is another picture. Again, this whole thing, it's nice when they label it. This whole thing is white pulp, and then this would be the central artery inside of it. Now, again, if you're new at this stuff, sometimes I see students mixing this up with like a Hassel's corpuscle from the thymus, right? All I will say is, again, this is not a great example, but usually the central artery will have a fairly clear lumen. It'll be hollow on the inside, whereas a Hassel's corpuscle won't be hollow, okay? And then last but not least, this is from your bones, from your spongy bone, specifically the stuff in the solid pink here, that's the actual bone tissue, that's the spongy bone. And then all of this junk that's filling in the spaces in between it, that would be red bone marrow. Okay. So once again, all of this would be the actual spongy bone, and then this material that's filling in the space everywhere, that's going to be the red bone marrow. Any questions about any of that stuff? All right, good luck. If you have any questions, shoot me an email. Please put the